Good afternoon, everyone. Thank you for the opportunity to talk today about investigations into BCL3 as a therapeutic target to treat prostate cancer. So my group research focuses on prostate cancer and how we can improve the treatment of advanced prostate cancer. So the prostate is a small gland of the reproductive system. It sits at the bottom of the bladder. Men with localized disease will typically receive surgery or radiotherapy. And despite some good responses, around a third of patients will actually relapse and need hormone therapy or androgen deprivation therapy. Localized disease has quite a good 5-year survival rate of around 98%, but as soon as castration-resistant prostate cancer arises, when hormone therapies fail, you get inevitable progression to metastatic disease where the 5-year survival is much lower. And overall, we see that prostate cancer is the second most common cause of cancer-related deaths in men, accounting for around 12,000 deaths in the UK annually, which is something that my group is hoping to alleviate by identifying new therapeutic targets for prostate cancer. So BCL3 is a transcription factor. It was initially identified as a non-canonical mediator of the non-canonical NF-kappa B signaling pathway. There's now evidence to suggest that BCL3 can modulate canonical and non-canonical pathways through P50 RelA and P52 RelB interactions, and that enables it to regulate key oncogenic events such as cell proliferation and survival, as well as migration and immune surveillance. But we know from over the last 20 years that BCL3 actually can coordinate. It's a master regulator of 5 transcriptional networks. That includes NF-kappa B, but also Wnt signaling, MYC signaling, JAK-STAT3 signaling, and SMAD3 signaling. And it does that through a variety of different mechanisms, through protein-protein interactions with other transcription factors or their co-regulators, or it can do it indirectly through protein modifications such as acetylation, phosphorylation, or ubiquitination, or binding nuclear BCL3 complexes to the transcription factor gene promoters to regulate these cellular events. So we know from human cancers that BCL3 is elevated in a range of different cancers, and this also includes prostate cancer. And interestingly, if we look at the frequency of BCL3 copy number gain and amplification, we can see that actually this occurs in around 4% of primary prostate cancer cases, but increases to 8.5 to 24.5% of metastatic prostate cancer cases. We also know that BCL3 is rarely mutated in prostate cancer, with around 0.1% of missense mutations identified, and that the protein expression is significantly increased in prostate cancer relative to the benign tissues, shown here in the Western blot of some different protein lysates from patients of either benign or prostate tumors, or across a tissue microarray where BCL3 is strongly expressed in prostate cancer tissue. So we've also been able to show a similar phenomenon in genetically engineered mouse models of prostate cancer, and that this expression of BCL3 does indeed correlate with increasing tumor aggressiveness. So a slow progressing tumor such as a PIK3CA mutant tumor has very low BCL3 expression, whereas PTEN deficient tumors, PTEN flox-flox tumors, or PTEN flox-flox in combination with PIK3CA mutation have a very sort of high level of BCL3 expression. We've also seen in the literature that if you knock down BCL3 and D145 cells, which is shown in A and B, the protein and mRNA level, if they implant those cells into immunocompromised mice, then you see a significant reduction in tumor burden. However, there's no change in the proliferative advantage here. There's no change in proliferation, but they do see an increase in apoptosis, suggesting that if you knock down BCL3, you can reduce tumor burden by increasing apoptosis. And they also showed as an aside that BCL3 is IL-6 regulated, and that was shown in normal and cancer cells. Over the years, we've been developing a BCL3 inhibitor at Cardiff. We've developed the first-in-class oral small molecule inhibitor, which was recently licensed to TNA Therapeutics, known as TNAT-101. It's an inhibitor of protein-protein interactions. It targets a unique pharmacophore in BCL3 to prevent it from binding with NF-kappa B transcription factor P50, reducing NF-kappa B signaling. And our target product profile has been quite good in showing that we get very robust PK/PD parameters, and our small molecule inhibitor offers a wide therapeutic window compatible with prolonged oral daily dosing in mice. So we hypothesized that BCL3 is required for prostate cancer growth and progression, and that our novel small molecule inhibitor TNAT-101 that targets BCL3 presents a novel therapeutic strategy for prostate cancer to improve patient care. And we're aiming to investigate how BCL3 contributes to prostate cancer progression and to determine therapeutic benefit of targeting BCL3 to prevent and treat prostate cancer. So firstly, we knocked down BCL3 in a metastatic castration-resistant prostate cancer human cell line called DUCAP, and showed quite nicely that we could get knockdown with 3 independent hairpins, and that correlated with protein reduced expression shown here in B. Then we do confluency assays in the IncuSite to show that proliferation is unaltered upon BCL3 knockdown, as was the viability, really supporting the previous data in the DUCAP xenografts where they didn't see any proliferative advantage or change. However, what was interesting was that when we performed low-density colony-forming assays was that all of the 3 independent hairpins were capable of reducing colony-forming efficiency, suggesting that TNAT-101 influences cancer cell stemness. We've also looked at the effect of TNAT-101 in this DUCAP cell line and show that again we don't see any change in proliferation or viability. However, this time when we performed the colony formation assay, we don't see any changes in colony formation. And these findings really indicate that regulation of stemness by BCL3 might not be associated with BCL3 P50 protein-protein interactions, which we know the TNAT-101 drug is targeting. Just to reassure ourselves that this wasn't a cell line specific response, we've actually repeated this assay in PC3 metastatic castration-resistant prostate cancer cells and the LAPC4 line, again showing there was no change in clonogenicity upon TNAT-101 treatment at 10 or 50 micromolar doses, and also in a mouse cell line of aggressive bone metastatic prostate cancer. We don't see any change in clonogenicity, suggesting that TNAT-1 cannot impair cancer stem cell function in vitro. We've also shown that BCL3 is a good therapeutic approach for targeting the prostate cancer, because if we knock out BCL3, we can see that there's no change to tissue architecture, and if we treat with TNAT-101, again, no change in tissue architecture of a normal wild-type prostate. We also think that this drug is particularly well tolerated. The data shown in C is actually from a triple-negative breast cancer patient, PDX patient model, xenograph model, and that shows that TNAT-101 treatment extends survival in this model relative to the vehicle. But if you look at D, you can see the percentage body mass, and you can see that there's virtually no change in the body mass upon treatment. So we believe that this treatment is unlikely to be harmful to wild-type prostate epithelium and is very well tolerated. So next we assessed whether TNAT-101 treatment was efficacious in advanced prostate cancer, and we treated the CUPCA1 PDX model and the PC2400 model. And you can see here that we saw very good efficacy in terms of reducing tumor volume, and at endpoint we see a significant reduction in tumor weight, suggesting that this treatment is efficacious against metastatic castration-resistant prostate cancer in this setting. When we explored proliferation using Ki-67 or apoptosis using cleavcaspase-3 by IHC and quantitate the IHC, you can see that we don't see any change in proliferation. And this time we also didn't actually see a change in apoptosis, which did surprise us a little bit initially. But this data was actually supported in the other PDX model that we ran, where proliferation this time scored using PCNA. Again, no change in proliferation and no change in cleavcaspase-3. So this data does sort of support the previous data that was in the literature, the DU145 xenograph model, where there was no change in proliferation. However, we're not seeing the same apoptotic response that was observed previously. to explore if the treatment causes initial proliferative or apoptotic responses, we have a model that's well set up in the lab at the moment for short-term explant treatments. So we can take proboscis P10 floxed mouse model of locally invasive carcinoma, harvest those tumours, embed them in agros, slice them on a vibrotome and place them on top of a gelatin sponge. And that enables us to do short-term drug studies. So if we treat for 48 hours with TNAT-101 compared to vehicle, we do actually see a reduction in proliferation at this time point. And when analysing cleaved caspase-3 immunohistochemistry, we see a surge in apoptotic cells, suggesting that this is an early event that perhaps isn't sustained over time in the long-term events. Interestingly, we've looked at REL-B here too to assess whether inhibiting BCL3 can impair the NF-kappa B pathway. And you can see here that the nuclear REL-B expression is significantly downregulated upon TNAT-101 treatment. And we're just working on the REL-A canonical NF-kappa B immuno now on explants. It's not been as easy to optimize that. However, we have managed to get the REL-A and REL-B immunos working on the PDX models. And again, we're not seeing any change at this later time point, again, suggesting that maybe this is an early event. And that an alternative mechanism is restricting tumour growth in the long term in vivo. So to look a little bit deeper into the mechanisms underpinning the efficacy that we see with TNAT-101, we performed bulk RNA sequencing on the PC2400 PDX tumours at the end point. Principal component plot shows very nicely that the drug in orange is clustering very nicely and the vehicle in blue is a little bit more dispersed but still separating away nicely. And the volcano plot showed quite clearly that we had 299 deregulated genes upregulated and around 40, I think it's 42 genes were downregulated. And the heat map indeed just shows the top 25 deregulated genes there. But to dig a bit deeper into the key pathway switches that are arising, we did gene set enrichment analysis and showed that actually there's only one key pathway that's deregulated, and that's called the extracellular matrix structural constituent pathway, suggesting quite a strong switch in the tumour microenvironment. And we saw quite a few extracellular matrix glycoproteins altered here, which we know can alter hypoxia, metabolism, immune modulation, and tissue stiffness. So we're quite keen to follow those up further. We've actually validated a couple of these and shown that the RNA-seq data is also correlative in QRT-PCR data, where we see an increase in tenacin C and fibronectin-1, which are all important extracellular matrix components. So we're now looking to see whether TNAT-101 can alter the tumour microenvironment to maintain tumour suppression. Another protein that popped out of this was the USP2 and CDKN2B. So if we remove all the mouse reads, we can actually look specifically at what's happening in the human cells. And that really pulled up to these, well, lots of different genes, but the two that we were particularly interested in were the USP2 and CDKN2B. So CDKN2B actually encodes P15 Inc4B, which is a known tumour suppressor. Which I think is super fascinating. It's a cyclin-dependent kinase inhibitor that was upregulated upon TNAT-101 treatment. But what we saw when we did the Western blot actually was that we're not seeing any change in the protein expression of this gene. We know that P15 forms a complex with CDK4 and CDK6 to prevent their activation by cyclin D1, prevents RB1 phosphorylation, suppresses G1 progression through the cell cycle. So it can cause senescence, and it's also induced by TGF-beta. So the fact that it's not upregulating the protein and only the mRNA might mean that an alternative growth stress driver is responsible for the overall tumour suppression. I'll come back to a bit in here. So what our working hypothesis at the moment is that TNAT-101 treatment transcriptionally activates a cell cycle arrest program whereby TNAT-101 potentiates anti-proliferative signaling as an early event. It could be by RB pathway, TGF-beta, or stress responses, and that then leads to a transcriptional upregulation of CDKN2B, which then causes growth arrest, and possibly induction of senescent-like state to suppress tumour growth. But I think that really the fact that P15 INK4B protein levels are not significantly changed upon TNAT-101 treatment could indicate that the protein is rapidly degraded, or that alternative growth arrest pathways are involved, and could indicate that TNAT-101 induced growth arrest is not driven by CDKN2B, possibly owing to global or selective translational repression, but we need to do some more experiments to clarify that. Also interested in USP2 is a deubiquitin enzyme that can remove ubiquitin chains to mediate cell cycle progression and metabolism, and also NF-kappa B and Wnt signaling. We know its targets include MDM2 and cyclin D1 that can promote G1S cell cycle transition, which could possibly indicate for early treatment resistance. But USP2 regulation can also restore homeostasis and trigger a stress metabolic adaptation response, which would sort of sustain treatment sensitivity as well. Again, ARTT-PCR validated the mRNA increase of USP2, but also similar to other proteins, we haven't been able to see any change in the protein expression. So we think that USP2 protein translation could be impaired despite the USP2 transcriptional regulation. There's several different ways this might happen. We're unsure of which one, but it could involve miRNAs, ribosome stalling, stress-induced phosphorylation of EIF2A, and it kind of supports the notion that there could be global translational repression owing to early senescence within these cells. We've also looked at Wnt signaling and shown very nicely that although Wnt signaling components are upregulated, Wnt target genes are not, suggesting that maybe the pathway is not activated. And that was really supported quite nicely in the CUPCO1 PDX setting, where we did immunohistochemistry for nuclear beta-catenin and saw no change. This model actually has a mutant beta-catenin hotspot mutation, so there's lots of nuclear beta-catenin here anyway, but treatment did not alter that with the TNAT-101 drug. And also no change in downstream targets such as CD44 at RNA or protein level. So we don't think Wnt signaling is relevant to support tumour growth in the PDX models. So just to summarise then, we see no change in some of the BCL3 transcriptional networks. So no change in c-MYC, no change in Wnt signaling, no change in REL-A or REL-B signaling. There could still be an early response, possibly, which we're going to explore, and that could also cause immune changes that we also need to explore. We do, however, see an increase in STAT3, suggesting that JAK-STAT3 signaling could be an important response to TNAT1 treatment to cause efficacy against the models that we've looked at. And we also think that deregulated TGF-beta SMAD3 signaling is also playing a role, and that could be via altered fibroblast phenotypes or abundance and the transcriptional induction of CDKN2B driven growth arrest potentially. So just to summarize then, BCL3 knockdown impairs cell clonogenicity in metastatic castration-resistant prostate cancer cell lines in vitro, but it does not alter proliferation or viability, which does actually contrast the TNAT1 treatment, which was ineffective against clonogenicity. TNAT-101 treatment is efficacious against the PDX tumours that we've explored in vivo and against an immunocompetent PTEN deficient high-risk localised prostate cancer ex vivo explant, suggesting it is a valid therapeutic approach. And we think that the efficacy is associated with growth arrest, CDKN2B dependent or independent, triggering early senescence that correlates with global translational repression potentially. And that's something we'll explore in more detail. And that BCL3 inhibition is associated with extracellular matrix alterations, indicating that TNAT-101 efficacy could be mediated by the tumour microenvironment, and that might be through BCL3 mediated functions in fibroblasts or immune cells. So in the future, we're going to be looking at the functional importance of BCL3 in the tumour microenvironment and the signalling roles of STAT3. So we'll look at fibroblasts and immune populations, look at the signalling events occurring, and we're also going to explore senescence, so SASP and other CDK inhibitors such as P21 and P16 to see whether a state of senescence has been entered into. upon treatment, and to assess whether TNAT-101 efficacy occurs in immunocompetent prostate cancer models like the PTEN deficient model in vivo in long-term treatment scenarios. And it just remains to thank everyone. Thank you very much for listening. Thank you for everyone that's done the work, and thank you to the funders. This work was primarily funded by Prostate Cancer Research UK. I'm very happy to take questions, but obviously I'm not here in person, so please use my email. It's at the bottom of this slide if you have any questions. I'd love to hear from you. Thank you so much.